8. In the light-dependent reactions, light was absorbed, H2O was split, those electrons were passed off to NADPH. Now in the light-independent reactions, which are called the Calvin cycle, ATP and NADPH, which were generated during the light-dependent reactions, are going to be used along with carbon dioxide ultimately to make glucose. This diagram represents the Calvin cycle. The Calvin cycle can be divided into 3 parts. The first is CO2 fixation. This is where carbon dioxide is actually captured. The second phase is CO2 reduction, and the last phase is the regeneration of ribulose bisphosphate. Ribulose bisphosphate is a 5-carbon molecule. A molecule of carbon dioxide is added to that, and that makes a 6-carbon intermediate. This 6-carbon intermediate is immediately converted into a molecule called 3-phosphoglycerate. That 3-phosphoglycerate goes through a number of reduction reactions. This is where both ATP and the NADPH are required. But following these reduction steps, a molecule called glyceraldehyde 3-phosphate is produced. This glyceraldehyde 3-phosphate is essentially 1/2 of a glucose molecule. So for every 2 turns through the Calvin cycle, 2 molecules of glyceraldehyde 3-phosphate are produced. Those can be combined to make a glucose molecule. In that last step, the ribulose bisphosphate is regenerated. So again, much like the citric acid cycle that we saw in Lesson 7, this cycle continues. This diagram just summarizes where these various reactions take place in the chloroplast. The light reactions that we saw, both the non-cyclic and the cyclic pathway, occur in the thylakoid membrane. This is where light is absorbed, excites some electrons. Water is split to replace those electrons. This is where oxygen is produced during photosynthesis. Also, during those light reactions, ADP is converted into ATP, and NADPH is going to be reduced into NADPH. Those both feed into the Calvin cycle that we just looked at, and ultimately the carbohydrate is produced.